everyone. Today we will be discussing, continuing with this optical microscopy. In the initial class, as I told you, just a recap, we discussed about the basics of the microscopy. We talked about what are the different types of optical microscopes. Then we talked about the mechanism of optical magnification of the objects. Then what are the different parts of the microscope? So we have covered. Then what is the resolving power of a microscope and how we can calculate it, that we have seen. Then we have discussed what are the different types of or the what do you call different parts and what they do and what are their properties of the different parts of the microscope, right? Then we talked about this how immersion medium they help us in improving the in capturing the images um, at higher magnifications. Okay, so we have told that we use some immersion mediums, and I also told that we basically use either glycerol or the cedar oil for the purpose, and they have usually high refractive index. Okay. So then we talked about the condensers, how they work and all these things. Then we talked about different types of cameras that are available. Then we also introduced the word trinocular microscope over here. Okay, and what is the advantage of the trinocular microscope over the binocular and the monocular microscopes? Then we have discussed about what is the importance of the inverted microscope and how it is useful in tissue culture examination. Then we have also discussed about this the reflection microscope, which are also regarded as the metallurgical microscopes also, and we have looked into the organization of the different components over here. Then we discuss about the difference between the bright field and the cross polarized microscopy. Okay. Uh, then we have looked into the functioning of the polarized light microscope. Then we have also discussed about the dark field illumination, then phase contrast illumination, how these four types of illumination systems alters the quality of the micrographs. Okay, we have discussed those. Then yeah, this is for the dark field illumination. We have discussed on these things, and we have also discussed how does the light moves through the optical system when we talk about the bright field system or the dark illumination system. Okay. So then, yeah, in the next part over here, we have discussed about how the dark field microscopy it works in the reflectance mode. Um, So this is a bright field objective and this is the dark field objective we have seen. Then we have discussed about the phase mechanics of the phase contrast microscope, how it works, and we have seen that the annular ring, it is made up of two layers. One is the retention layer and one is the gray filter. The gray filter, it dims the light. That means it reduces the intensity of the unrefracted radiations, whereas the retention filter, what it does is that it changes the phase of the light. Okay, so due to the combination of this retention filter and the gray filter, we have a phase difference and a reduction in the intensity level of the light of the unrefracted light which passes through this phase ring. Okay. Then we talked about different types of contrast mechanisms in refracted microscopy. I for this one I told you that we'll be discussing about this DIC microscopy in the transmitters mode, and then we can look into the thing. So if you see light through here, it goes through the polarizer 45-degree angle. So the electromagnetic radiations which are being generated, they are having an orientation of 45-degree. Then it passes through the Normansky prism, where there is a this Normansky prism. It what you call divides the polarized light at 45-degree into 0-degree and 90-degree polarized light, and also they have a shear difference and phase differences over there. Then it passes through the samples, and then you have the objective lens. It collects both these 0-degree and 90-degree polarized light, and then again it goes through the Normansky prism, and then using cross polarizer over here at which is present at 135 degree, we block the polarized light at the 45-degree angle. So at the other angles, we are having the radiations which can be collected. Okay. So in this way, this transmitted DIC microscope it works. However, if you look over here, you see the polarized light is there. The light passes through the beam splitter. It goes through the Normansky prism. So there will be splitting of the rays. They will come. They will interact with the samples. Okay, with the specimen. Again, after getting an interaction over there, what it will do? It will move up both the. They will move up over here, and again they will be recombined using this Normansky prism. So over here the same Normansky prism, it is what you call it is decomposing the light, and at the same time it is again reconstituting the light. Okay, or it is what you call it is again merging the light, the differently polarized light. Okay, then this combined light, combined polarized light, it goes through this beam splitter, then through the analyzer, which is 135-degree angle system. So this is the 45-degree polarizer. Again, only the light except 45-degree polarized light, it passes through this analyzer, and this light goes through the tubeless. The rest of the mechanism, it is the similar. Okay, so now you can correlate between the transmittance light DIC microscopy and this reflected light microscopy. Now if you see, I told you that the contrast mechanisms in reflected light microscopy we have discussed over here. Similarly, if I give you a question on discuss about the contrast mechanisms in upright microscopy, then what you will be discussing? The normal bright field microscopy you will be discussing. Then you need to discuss about the dark field microscopy, then polarized light microscopy, DIC microscopy. Apart from this, you have to also discuss about the phase contrast microscopy. Okay. So in reflected light microscopy, we can increase usually by three ways. So B, C, and D. And how it improves the contrast, you need to discuss this over here. However, in the upright microscopy, there are four ways of improving the contrast of a microscopic image. Okay, so this you need to keep in mind. We discussed about this stereo microscope. Then we also discussed about the fluorescence microscope. Then we talked about fluorescence merging. What is fluorescence merging? I told you that by employing a set of emission filter and an excitation filter over here. So suppose there is an emission filter, excitation filter EX1, and say emission filter EM1. Say for example this combination is working for the blue channel. Then similarly for EX2, EM2, it would work for the green channel. Then EX3, EM3, this combination of the filter, it would be working for this red channel. Okay. So after you have clicked all these three images, we then go for the superimposition of the images one over the other by the digital methods, and this process it is called fluorescence merging. Okay. Now coming to the confocal microscopy, confocal laser scanning microscopy, in the this fluorescent microscopy, if you see, we were using a light source which may either be a halogen light source or it may be a LED microscope. LED light. Okay, so either halogen light or the LEDs, they can be used as a light source, and then we have the excitation filter and all these things. However, in the case of confocal laser scanning microscopy, we use a laser. Okay, why we use a laser? We use a laser because laser is having a high power. Laser is having high power. Okay, now if you see all the things, you will that it is similar to your fluorescence microscope. Okay, so if you see there is a filter over here which will allow you to filter out or select a monochromatic light. Even we can use the laser source which is having a very narrow band over here. Okay, so the laser source in itself, it is a monochromator. It is a laser light is a monochromatic light source. Okay, so it may be condensed, or if you want that you want to use a filter, then you may use a different a kind of filter to allow a certain band of light to pass through. Okay, so so once you are getting this thing, once the light passes through here, that dichroic mirror is there, then the light is being reflected through the objective onto the sample, and then it is getting reflected back through this objective, and again there would be an filter over here. This filter is again the emission filter, EM filter. Okay, then if you see there is a pinhole which is an additional stuff. So yeah, there is another scanning XY control. So if you want to move your objective through X and Y direction, that means across the surface of the thing, you can go for the this XY control system. However, we will it is just like suppose you are having this sample surface. Now you are looking at this position. Somehow you are interested in this position at this position. So you want to bring your microscope to that position. Only then we use this XY control. The So let us forget this thing. That now let us concentrate only on the optical path. In this optical path, as I told you, there is only one additional thing which is there, and that additional thing is the pinhole. Now what this pinhole is doing, if you see, when we are focusing a light onto the sample, since the laser it is having a high power, it has the capability to even penetrate to a certain distance within the sample. Okay, now by moving this setup up and down, we can bring, say, this dark line over here. So at this plane, our light or the microscope, it is in focus. Okay, so what this pinhole is doing is that it is only allowing the light from this focal point to pass through this pinhole. Okay, now in this case, in the line where this dotted line, the light gray dotted line, if you see the out of focus line, it is here. Now it is not allowing the out of focus lights to pass through this pinhole. It is, this pinhole is blocking. Okay, so in this way, this confocal laser microscope, what it is doing, it is allowing to get the information just from one single plane. So your depth of focus in this case would be narrow. However, since there are no arrangement as such in the fluorescence microscope, whatever the fluorescence activity you are seeing, it would be through a particular region, or the through the particular thickness of the sample, until where the light is able to penetrate. Now please remember, we can also design the fluorescence microscope in the transmission mode also. I will discuss about that a bit later. Okay. Now, if you see over here, now if I want to get the information about this out of focus layer, what I need to do? I need to bring down this pinhole at this position. Okay. Then what will happen? If you see in the previous case, what the light rays which was in focus, it is now getting blocked. Okay, and we are only getting the out of focus light into the system, and if we bring this pinhole downwards, we will be able to get an information from within the within the samples. Okay, so in this way, just by moving this pinhole, we will be able to get information about the different layers within the sample. Okay, I hope all of you have understood this thing. If you have any question, please let me know. Okay, now coming to the fluorescence microscopy, I told you that we can design this fluorescence microscope, in the bright field or in the upright microscopy also. So what would be the uh, what do you call what would be your block diagram for that? The block diagram would be, you have the light source over here, then from the light source you will be having one excitation filter. So you will be having the excitation filter, EX filter. Okay, then this excitation filter, the light coming out of this excitation filter will be collected by the condenser. Okay, so this is the condenser. Okay, now then you will be having the sample stage. You're having this sample stage over here. Okay, now the light, it will pass through this sample. Okay, now the problem over here is that you will be having two kinds of light. One is the light which is coming through this excitation filter. Okay, and the other light what you will be getting is the fluorescence light, which will be generated in all the directions. Okay, then we will be having the objective lens. Okay, now after this objective lens we will be having this optical tube. Okay, now within this optical tube we will be having a what you call emission filter. Okay, now this emission filter should be blocking the excitation rays, EX rays. Okay, so in other words, you can use a, uh, a band stop filter also, so that the emission ray is blocked. Over here also, if you see, I told you in my last class that this emission filter should be such that the excitation rays which will be going back again through the reflection mode towards this emission filter, they would be blocked. The same thing would happen over here also. You should have an emission filter which will block the excitation rays, and then you will be having this ocular eyepiece. Or instead of this ocular eyepiece, you can have a detector. Okay, and if it is a trinicular this thing, you will be having an dedicated port, something like this. So it would simply look like your any trinicular microscope over here. we have discussed now, right over here. So what we will be having is that in these cases, usually what happens in the transmission microscopes, you have a filter over here. This is your excitation filter, and then if you see, there is an attachment which is made over here at this level where we have the emission filters. So an attachment is made over there. So if it is a triangular microscope, you can use both the eyepiece ports over here to visualize the fluorescence activity. Otherwise, and you can use this C-mount port for the capturing of the image. Otherwise, you can attach this what you call cameras into the eyepiece of the microscope. Okay, so it may be the fluorescence microscope, it may be a what do you call it may be a binocular microscope, it may be a monocular microscope, and it may be a triangular microscope in the upright formation and even in the reflected formation. However, since fluorescent microscopes they are bit costly, usually they come either in the binocular or in the triangular format. Okay. So with this, I complete the chapter. However, I would like to discuss about one of the work which one of our student he has developed. Uh, he has developed a multimodal microscope system, and over here this is the block diagram of a multimodal system. So if you see, it is having the several systems. So this OLEX is basically open loop electrical control system. What it does, it controls the movement of your optical system. And usually you have this SIA. What is SIA? It is a sample illuminator assembly. Then you have FHA. It is a holder as filter holder assembly, which is basically associated with the condenser. Then you have the sample, then you have the objective lens and the eyepiece camera, and these together it forms the SMIA along with the optical tube. And what is SMIA? SMIA is sample magnification and imaging assembly. Okay. Then from here we have this information going to the Raspberry Pi camera, Raspberry Pi which is a microprocessor which computes the images and helps us to take the image. Okay. Now, what is the advantage over here? The advantage over here is that if you we can use a series of filters over here, which can allow us to take different types of images. Okay. The first one, it is called the this dark field illumination filter. So if you see the central part of the light, it is being blocked. Then we have Reinberg illumination. This B and C, they are regarded as Reinberg illumination. In Reinberg illumination, usually the central part of the filter, it is made slightly darker than the outer layer, and you need to have a contrast, a different contrast color over here. If the contrast between the both the colors, inner and the outer colors, they are similar, then you will not get good results. Similarly, we have used another type of Reinberg illumination filter, and then we have used one this what you call this oblique illumination filter. Oblique means oblique illumination filter. Okay. Now, what is this and why you want to use that? Now, suppose you are you're looking into the light source over here. So I'm looking into from the top view. So from here the light comes. Now, when you place this filter over here, okay, let me use black color system. Now see, it is a rectangular part. Okay, so this is the darkened part and this is the open part. Now as you move this filter over the illumination system, so what will happen as you go towards, say, this is position A and this is position B. As this position B is towards this optical this light source, you are seeing that only a certain part of the light would be passing through. Just, sir, class নিচ্ছি পরে কল করছি. Okay. However, when you move this filters towards the B, that means this area near to the A, it would be in it would be over this overlapping over here. So a large amount of light would be passing through. So what is happening basically? So till now, if you see, when we are using the light, using the light source, we are illuminating the system in through all the direction, except in the dark field where we are stopping this central part of the light. However, in this case, we are blocking the light at a particular angle. Okay. Now, when you move this and when you look into the system, so what is happening? Suppose you are having this object over here. Okay. Now you are saying that you are putting the light rays at a particular direction. Okay, so since the light is coming from this particular direction, the other side where there is no light, it will be appearing somewhat darker. So this oblique illumination system, it would give you an image, or it would give you a perception of a 3D image to your objects. Now you would say, sir, we talked about DIC yesterday. It was also doing the same thing. So why we need to do this? The thing is that when you try to go into the DIC microscopy, it would cost you in lakhs. Okay. However, if you use this oblique illumination filter, it would give you a similar effect. Okay, it would give you a similar effect, but it would give you a 3D effect even at a very low cost. Okay, so when you come to the lab, we will try to show you the things, how the things work. Okay, then we have this polarizing filter. It can also be used. So these are the filters which have been developed previously. Now this is the microscope which it has been developed. If you see, there are these joysticks which are basically the control system. Okay, then you have a condenser over here. Since everything is black, it is not visible over here basically, and at the bottom you are having the light source. If you see some bright spot, it is coming, and then just over here we have the filter holders. Okay, see this image. This image is basically giving a 3D perception. So this is definitely a over here we have applied the this thing oblique illumination filter. Okay, see, so we have analyzed some samples, and if you see, so this is the analysis of a curved sample which has been taken by the microscope. So this is the normal bright field microscope A. Second is the oblique illumination microscope. If you see over here, and if you compare A and B, if you see, you are getting a 3D like effect over here. Okay, however, it is since the size of the micrograph is quite small, you are not able to look into the effect to the greater extent. And the last one is the C1. It is polarizing microscope. So in the polarizing microscope, as I told you, during the cross polarized light, everything will be So it gives dark and we are getting almost nothing. Okay. Now the next one is dark field imaging. When we go for this dark field imaging, if you see your images, it is basically appearing as the foreground, it is appearing as bright, whereas the background it is appearing as completely dark. Then the second one is dark field imaging with your oblique illumination. Okay, so if you see over here, so if you would see, you would get that here we are getting some 3D effect. Here also, if you see, you are getting 3D effect. So if you see, there is a slight shadow over here, and there is a brighter region on the other side. Okay. In the polarizing sense, curd, it is having a lot of proteins and all these things which do not have crystalline properties. So we are not seeing the polarizing effect over here. In this row and the next row, we have the rainbow illumination system. Okay. So if you see the rainbow illumination over here, blue and yellow. If you see blue and yellow, getting the blue which was at the center, it is getting the background. That means if you compare the dark field imaging and over here this rainbow illumination, in dark field imaging the central part it was blocking, so no light was coming through. However, when we have changed this thing, we have used a colored filter, the dark area in the dark field imaging, it is getting the central part of the thing. So in the bright field imaging, we have the all the central part, it is having the white light usually, so we get the white background. However, since in the rainbow illumination, since we are having a different color system at the central part, so the image background it is acquiring that blue part. Okay. Similarly, over here also it is acquiring the blue part, and if you look into the this foreground region, if you see, it is appearing a golden color thing over a background of the blue system. Even though our system does not have a color information over here, except this one small microbe, which was due to the presence of the lactobacilli. Now, if you look into this image, blue background, yellow this thing with your oblique illumination, if you compare the quality over here, the contrast of this image, it is quite high. Plus, it is giving us a 3D effect over here. Okay. Now, the advantage of such system is that when you try to develop such multimodal microscopic systems, you would be able to generate a lot of microscopic microscope or micrographs, not this microscopic, the micrographs which is having different properties. For the same region, you can see we have generated 12 types of images, and all these images they are having their own beauty. Okay. Next, let us then we use the dairy milk chocolate which is available. Now, if you see over here, the 3D effect is much more visible because the microstructures are quite good over here. In the previous case, it was not so visible over here because we have compressed the picture. Now, if you see, the 3D effect is much more visible. If you see, there is a shadow, and here there is a brighter spot over here, right? Now, in the polarized light, if you see, you are having birefringent activity. What is birefringent activity? The birefringent activity is the ability of the crystalline material to produce different spectral components when placed under the polarizing system. Okay, so if you see, you are having multi-color system, and these systems are these may be due to the presence of the sugar crystals. Second, it may be due to the fat crystals which are present within the molecules, right? And the same things, if you see, over here also. So this is the bright field. So this is complete bright field. This is your oblique illumination, and this is the polarizing illumination. Okay. Similarly, this is for the dark field illumination. Now, if you see in the previous slide, our background was quite dark. However, over here, if you see, while analyzing the chocolate, the dark field illumination, did not have much effect in this case. Okay. Then this was dark field plus oblique illumination. Then dark field plus polarizing illumination. Here Reinberg illumination, then Reinberg illumination plus oblique illumination. Here Reinberg illumination plus polarizing light or polarizing information. Okay. And lastly, if you see, we have another Reinberg illumination, say Reinberg 2. So Ri2 plus oblique illumination and Ri2 plus polarizing information. Okay. So if you see, just by playing with the filters, if you are having a particular scene, how we can alter all the stuff. Another important thing which I want to show over here, we got some wastewater which was being cultured at Professor Angana Sarkar's lab. So she works on environmental biotechnology. So she was quite helpful in providing us with the sample. Now, if you see over here, when we analyze the image or micrograph in the bright field microscopy, we did not get any particular information. However, as soon as we applied this oblique illumination, if you see these globular structure, 3D like structures, they were quite visible, and they were not visible over there. Okay. Now again, if you look into the dark field illumination, again over here in the dark field illumination, we have been able to extract some information over from here. Okay. Now in this case, where we are having the Reinberg illumination plus oblique illumination, again if you see, we are getting a blue background, which is for the inner blue color which we have used. However, if you see over here, what is happening, we are getting some 3D information. So these samples, they are so small that they were not able to pick up the information from the outer green light positions. Okay. Now, if you look into the Reinberg illumination 2 plus oblique illumination, where we had blue inside and yellow outside. If you see the larger clusters of these microbes, they were able to pick up the yellow color. Okay. Now again, see, in all these cases, in all these types of micrographs, we have taken the image from the same scene. What we have changed? We have just changed the the filters that we are using, and by playing with the filters, we have been able to get different types of results. Madam, class আছে পরে করছি. Okay. Now this is an oleogel sample which was prepared by one of our PhD students. It has fat crystals. Now see, it is giving us the beautiful needle shaped crystals, and here also you can see the 3D structure is quite visible, which was not this what you call the crests. They are quite visible over here, which was not visible in the bright field microscopy and also in the polarizing microscopy. And these crests, if you see, they are visible everywhere, over here also, and here also. And if you see the yellow color system, dark blue plus yellow color system, if you compare the results, you will find that it is giving a better result than the Reinberg illumination what we have used in this third row. Okay, so with this I conclude this microscopy lecture.